First, I would like to thank the organizers for this great opportunity to present our results devoted to the computational characterization of biosynthetic route to the one of the most abundant neuropeptides in the mammalian central neural system that we have recently made in our group in the laboratory for quantum chemistry and molecular modeling at the university at Lomonosov Moscow State University. So the N-acetylaspartylglutamate is the most abundant peptide neurotransmitter that acts actually as a retrograde neurotransmitter. It is packed in the synaptic vesicles. It is released upon the calcium ion entrance in the presynaptic axonal terminals, and is metabolized in the synaptical cleft. So the most important information about this neurotransmitter is that it acts directly as an agonist on metabotropic glutamate receptors type 3, and it is really selective agonist with the effective concentration, half effective concentration in the range of tenths micromolars. But actually there is information about it acting on NMDA channels, sometimes as antagonist, sometimes as agonist. It depends on the pH and subunit composition of this channel with this diabolic effective concentration. Also it is included supposedly, so it is included in the development of schizophrenia, and was stipulated to have implication in pain, epilepsy, ischemia, traumatic brain injury. Also, one interesting fact, it is successfully commercialized in Portugal as anti-allergic drug that acts as a mast cell membrane stabilizer with the patented name NAEBEC and international non-patented name spaglimic acid, and it is sold with the price of 12 euros for 1 gram in the water solution. Okay, metabolic pathway for NNAG biosynthesis in brain proceeds through two steps. There are two enzymes, one synthase and one synthetase, NAT8L and RIMCL-A, that we are modeling in our group. We have built a model for NAT8L that is a membrane-attached protein, albeit very well characterized experimentally. Two of these enzymes are very well characterized. As you can see on the slide, we have extensive information about thermodynamic and kinetic parameters. So we can make use of the fact that there is none crystallographic structures available for these proteins to make our models starting from primary sequences and proceed to the study of their dynamic properties in the hope to understand the mechanisms how these enzymes work. As probably you all know, the most useful method to predict the structure of proteins now is AlphaFold 2. After its success, it is widely recognized. But should I put it as much simple as I can? It starts with searching databases of protein sequences and to find the most similar and align them. And after that, multiple sequence alignment of the protein sequences is used to extract the coevolutionary information. Coevolutionary information can be used to predict distances between residues in the amino acid chain, and also can be directly used to predict the structure of the protein. The structure of NAAG synthetase that we obtained through two different methods, namely through homology-based modeling and AlphaFold 2 pipeline, are presented on this slide. You can see that RMSD is 3.5 angstroms, but all these structures are of the upper form of the enzyme, so we have to put in the ligands, the substrate, and cofactor. Cofactor here is ATP, just because it's ATP grasp domain. So we put in ATP, put in magnesium ions, and intermediate acetyl phosphate and glutamate. After that, we can start dynamical exploration of dynamical properties of the enzyme. If we can see that RMSD on the trajectories is also in the range of 3.5 angstroms. And albeit the RMSD is stable across these trajectories, we can see different outcomes. In one of them, for example, we can see a large movements of the loops that are gates for the active site. In others, we can see that active site is stabilizing. So I think the main idea here is that we have to use large computational facilities to in order to proceed with molecular dynamic studies of enzymes. It is indispensable, after all. But the most interesting aspect of enzymes is in their active sites. We propose here the mechanism for NAAG synthetase. The mechanism consists of two steps. And at the first step, we can see the acetyl phosphate intermediate formation. And on the second step, we can observe the attack of nucleophilic amino group of glutamate, obviously after the deprotonation, on the electrophilic center of acetyl phosphate intermediate. All of this leads to the formation, to the synthesis of NAAG. We also studied the system with the molecular dynamics using hybrid molecular mechanical potentials. The information about the composition of the quantum subsystem you can see on the slide. The most important result of this endeavor was to observe the stability of the distance between the nucleophilic nitrogen and electrophilic carbon across some of the trajectories. We have in total 15 picoseconds, and 1.5 picosecond per day was the efficiency of our calculation using the interface between NAMD and TerraChem software that was already presented by Maria. So we can say that the models that we obtained can be used to guide, for example, mutagenesis studies, and also we can propose the mechanism for this enzyme using our trajectories and our data. So thank you very much for your attention. I'm ready to answer your questions.
Thank you very much, Artyom. I don't see any questions in the chat, so maybe somebody wants to ask it by voice. So while the others may prepare, I had some question to you. So just a technical question. What so as far as I see, you use DFT functional for quantum part?
Yes, we used PB0 hybrid functional with the 3 dispersion correction, as you can see on the slide.
Okay, I see. Yeah, I haven't noticed it. Yes. Okay, but have you ever observed that, for example, that functional may like have a different influence on the, so the conclusions would be different if you use different functionals. For example, here you say that you use D3 dispersion correction. For example, if you will switch it off, will something be changed there? Will it be really poorly displayed what happens with a protein or with this enzyme?
It's a really interesting question. I think we could just switch it off and rerun the calculations, but I think personally, I think that use of the D3 correction is just routine thing in such calculation. It's just useful. It was shown to be useful in some model systems. So probably turning it off could have any possible could result in any differences, but I don't really know now. It is so or not.
My question is arising from the, so actually dispersion is so it's long range correlation. So this D3 correction is just used for the description of this dispersion term. So in peptide, probably dispersion is really important, may be important, but I'm not sure it will be important for the quantum part of, so in where the reaction itself is goes. No?
I think that inclusion of this correction is dictated by the functional, just because it cannot correctly describe it. So why not using it? But
it's kind of fashion as to me. so now everybody asks for including dispersion correction. When I was young, nobody used it because it wasn't invented. Okay, we have also some questions from the first question from Igor Polakov. Are there any active site rearrangements switching from classical to quantum MD?
there are the rearrangements in the rotationary states, states of arginines that coordinate the active site. These motions were observed in our QMMM trajectories. They are not very large, but they are here.
Okay. The next question is from Dmitry Shulga. What is the main benefit of using QMMM for this system? Are you going to study the kinetic parameters of the reaction next? In the latter case, it is understandable.
Yes, certainly we could start and proceed to describe the mechanism using these hybrid potentials. Yes, it would be useful, very useful, to understand the kinetic aspects of the reaction.
Okay, and so again, I have just to add to Dmitry question is there like plot how well the kinetic parameters that you can calculate from this enzymatic reaction, how well they correlate with kinetic parameters which are observed experimentally?
Yes, talking about these two enzymes, this synthase and synthetase. Here, I can see that our preliminary results for N-acetylaspartate synthase are for kinetic constant, catalytic constant of this enzyme are something better than those obtained from the experimental investigators. Probably because of the fact that it necessitates solubilization of the protein, of this membrane attached protein, that can result in something worsening of its catalytic functioning. But that is speculation. So we can say that. So we can say that it is a very difficult endeavor to understand and to compare sometimes, and to compare theoretical results and experimental results. But possibly and hopefully there are some explanation for this.
So you can use a Eyring equation, for example, for calculate rate constant directly from delta G of activation here?
Yes, obviously.
Okay. So we're about to finish. Okay, thank you, Artyom.
Thank you very much.